Okay, here it is. This is it. Last level of DNA sequencing. It's called nanopore sequencing. The company that's out in front right now is called Oxford Nanopore, which who cares? But the stupidest thing is they named their things by with ion, and so they have like the grid ion or whatever. But the smaller one is called the min ion. Yeah, spelled just like minion. They named their new cutting edge technique after minions. And if that's not trolling, I do not know what trolling is. Yeah. So, fine. Go out and invent something. Pollute the world with stupid ideas. It's fine. So, nanopore sequencing, there's a nice video right over here, and go watch it later. Let's talk about it first, and then it'll click and it'll make sense when you watch it. Or watch it first, then let's talk, and then, yeah, watch it again at the end. And then you'll be like, oh, I get it all. Yeah, the world is open to me. Yeah. So, nanopore is a little tiny pore, and the DNA goes through the pore and gets sequenced as it goes through the pore. So basically, all you need is some kind of little hole of consistent size and a way to push or pull the DNA through that hole. Yeah, like spaghetti into a toddler's mouth. Slurp. And there you go. You will have nanopore sequencing. Yeah, sounds so stupid. You know, why didn't I invent that? Why didn't you invent that? Oh my gosh. Well, it's already invented now, so too late. But, next time. Okay. What kind of pores can we use? Whatever you want. The pore just has to be consistent in size so that always there'll be a certain flow rate. And you want it to be only just bigger than a strand of, like the diameter of a strand of DNA. Now, DNA has 4 nucleotides. You may be surprised to learn. Yeah, wow, I hope you were sitting down for that. The nucleotides are a different size. And so what you're doing is you're pulling these things through. Each one blocks the channel to a greater or lesser effect. Yeah, or greater or lesser degree, I guess is a better way to say that. And so you can make. There are people that are doing nanopore sequencing with little holes in graphene layers, and that works. You just need a hole that you can push stuff through. Proteins are commonly used because bacteria make a lot of proteins that happen to be, they're like transporters. So they have a little hole in the center, and the hole is always exactly a particular size, and they're used to being embedded in a membrane. So if you lay down a membrane, you can just put them on and they will embed themselves right side up in the membrane. Very convenient if you're building, assembling lots of these. So yeah, that's what people tend to use, but you can do it with a lot of different ways. All you need is a hole. That's it. Yeah, you're going to slurp that DNA or RNA chain right through it. So the next step in the technique is that there's actually a problem, which is that if you're trying to push DNA through this hole, it goes too fast. It goes actually faster than we can reliably measure. And if you can't measure it, then you can't really read the sequence because you've already slurped the entire spaghetti noodle before, you get the first readout. So we need something to slow down the speed at which these go in. One of the ways to do it, and there are many different ways to do it, but one of the ways is to put a protein on there like a helicase, and that will actually feed the nucleic acid in. At a very reproducible, reliable rate that's not too fast. And so helicases, yeah, you probably have heard of helicases like, oh, they unwind double-stranded DNA. Yeah, like so. Like, yeah, but what a helicase really does is it moves along a strand of DNA or RNA, but let's say DNA, and it happens to unwind anything it runs into. It's going to use ATP to actually power that movement. So a helicase will just move down a piece of DNA. Or, now here's next level stuff. Imagine the helicase can't move. The DNA will just be pulled right through it. And so a helicase will feed DNA in one direction using ATP. Yeah, reliably, reproducibly. And so one of the big companies uses helicases for some of its applications. But like I said, there's a lot of ways to do this. That's just one of them. But yeah, so it's a controllable way to slowly, it's still pretty fast, but to slow down the rate at which DNA goes into the hole. Okay, then, all right, you're like, yeah, I'm feeding this through the hole. How do I actually tell? And you tell with voltage differences. You're like, but the nucleotides don't really have particularly different charges. And you're right. They don't really have charge at all. There's a -1 on the phosphate backbone. And other than that, yeah, the side chains have a little dipole moment, but they're not charged properly. They're just like an uneven distribution of a neutral charge. But the way it works is that you have a whole bunch of ions that are lined up here. You can't see them because they're tiny. Yeah. And you're going to set up an electrical field. So you're going to have a voltage difference, like in a battery or like in electrophoresis, exactly like in electrophoresis. Instead of pushing a piece of DNA through agarose gel, for example, or polyacrylamide, you're going to push it through a little tiny nanopore hole in a membrane. Yeah. And so there you go. You're not slurping it through, you're pushing it through, which is a hard way to eat spaghetti, I gotta say, but works well for sequencing. So what you're measuring is how many of these other little ions are going to go through. So the DNA has a negative charge on its backbone. RNA does too. You can sequence RNA directly with these techniques. But there's going to be a lot of other little minus charges. So think little chloride ions, something like that, or OH minuses. And they're all going to be being pushed in the same direction as the DNA. But as the DNA goes through this very narrow part, it's going to block the flow of the other ions. And so a little DNA nucleotide, like a C or a T, is only going to block it a little bit, and a big nucleotide like A or G is going to block it a lot, and so you're going to get very little ion flowing in for just a second while G is in the channel or A is in the channel. And actually there's a difference for each of the 4 nucleotides. You get a different amount, and it looks kind of like this. So we've applied our current across this. We've got our voltage difference on the 2 sides. And so we will measure the ionic strength of what is coming through the pore. And so when a T is blocking the channel, it blocks most of it, but not all. So G blocks this much, it's like a ton. A blocks that much, and C doesn't block very much. It blocks just up there. And so, yeah, depending on which nucleotide is right there squeezing through the little hole, it's like squeezing into a turtleneck sweater. It takes a minute to get through. And so you get a temporary pause. And like with a C, you can get some ions passed, but not very many. With a G, you can basically get no ions passed while it's blocking. It's a complete block. Yeah, it's like putting on a too small turtleneck sweater. And they go through in a particular order. And so you know the order of the nucleotides and what the nucleotides are. That, my friends, is sequencing. Yeah, that is nanopore sequencing or pore-based sequencing. Yeah. And then, all right, so the ones who've had a physics class, whatever, this is for you. I don't really understand this. But this is basically the setup for how can you read whether or not there's a difference in membrane potential, either side of a membrane. There are other ways. It would be similar to what neurobiologists do to measure the transmission of a signal up a neuron. It's called like a patch clamp technique is the one they always cite. And so there are ways. This is one way. You can have a little droplet that's on the other side, and as more ions come through, you can actually read out basically the strength, the ionic strength, the charge in that little droplet of water. So this is one way to do it. The ways that the main companies do it are all totally proprietary. You cannot find them very easily online. And they're different, and honestly, it's just like electrical engineering at that point, and that's a different class. Let those guys earn a buck. Yeah, we've done all the hard work here. So there you go, nanopore sequencing. Now, if you watch or rewatch the video, it's going to make a lot more sense, I think. But yeah, you just slurp a bit of DNA through a tube. And there are lots of little refinements on this that each of the companies use, but the main technique. That's all you got to get. And so, yeah, with Nanopore, each pore can read out one DNA sequence, and then another, and then another, and then another, and there'll be a little gap in between them. And so you can generate lots and lots of very long reads, because the pore, it can slurp an unlimited size piece of DNA through. And so it can read you 30,000 bases all at one time, whereas we were getting like 50 to 250 with pyro sequencing. We were getting up to 1,000 with Sanger sequencing. But yeah, tens of thousands, no big deal. It makes some mistakes. It's a little less precise than the other sequencing techniques, but you just resequence the same thing, take the average. It won't make the same mistake every time. And yeah, you get a real good, clean sequence very fast. Yeah, so there you go. Nanopore sequencing, not so bad. All right, you made it to the end. Now you know about how we get all this DNA. That's good. The next parts we'll talk about how we get some other kinds of information, but that's enough for now.